Okay, this is going to be our RNA processing assignment. And so the first drawing is to draw this image, not in this detail, and using the terms that I'll include here. And so let me just add a drawing pen. Okay, so starts out with, we're going to draw a cell with a nucleus, and then we want to draw, what happened here? Then we're going to draw inside here the gene transcription. So that's what we've studied a lot. So you can draw a gene like we like to draw and label it and show a transcription factor activating in it and show some RNA being made. That RNA that gets made is called the primary transcript. So that's what we have learned about so far. And it's in the nucleus when it first gets made, and then RNA processing occurs, and what leaves the nucleus is the mature RNA. And so we're going to talk about RNA processing, and RNA processing occurs in the nucleus, and it takes the primary transcript to the mature RNA. As a side note, I should just put over here that remember that for genes, we have two categories of genes. One category of genes are called coding genes, and they code for proteins. And so we'll especially be focusing on here, on this, in this first drawing. And then we have non-coding genes, and we want to talk about those some as well in this assignment. And so non-coding genes are simply genes that don't code for proteins. They just code for mature RNA, and it could be like tRNA or ribosomal RNA and small nuclear RNA and so forth. Okay, so all genes get transcribed into RNA as a primary transcript. That primary transcript goes through the RNA processing. So the handout that the assignment we're working on is going to be stuff that happens inside the nucleus, and then the result of RNA processing is a mature RNA that leaves and goes into cytoplasm. And so we'll be learning about the rest of this pathway as we go along. So let's just include this in the first drawing to keep track of where we are. We are, in this assignment, working inside the nucleus. Outside of the nucleus, in the cytoplasm, if it's mRNA, that's messenger RNA, that's from a coding gene, it's going to code for a protein. So that's what we'll follow through on here. And before translation occurs, there is this option where some RNA can get degraded. And that's in a process called RNA interference, is the main process that we're going to learn about, and can include, and we'll include that in this RNA processing assignment, even though it happens outside of the cytoplasm. So it's not a process, it's not an RNA processing step, but we'll include it here. And then there's another type of RNA interference that blocks translation. So we'll also learn about that here a little bit because it's just the best time in our course to learn about it. So tucked into this assignment that's happening here, RNA processing, we'll also follow the mature RNA out to the cytoplasm and learn some about RNA interference and the two categories of RNA interference in this assignment. Okay, so then translation is going to occur, and there are ways for RNA to get degraded that's not RNA interference. There are ways for translation to be blocked that are not RNA interference, but those will be our favorite examples for this course. Great. And so then we're going to have the protein get translated, and now it's a protein, and we can have post-translational regulation. And we'll talk more about it in our course, but just to put it all together in this assignment, post-translational regulation occurs things like binding to 14:3:3, and that's going to affect it, or degradation of the protein. We've seen examples of that. And in post-translational regulation, a subset, some types of post-translational regulation are called post-translational modification. So distinguish between those two terms. A modification is a covalent change to the protein. So, for example, FOXO gets phosphorylated, and that's a post-translational modification of it. FOXO binds to 14:3:3, that's a post-translational regulation. So the big group is all examples of regulating a protein after it's been translated, regulating its activity, and some of those are by modifying the protein, okay? And we want to go all the way here because we're interested in gene expression. And in our course, you said, is the gene on or off? Well, it turns out the gene could be transcribed. It could be on, and any step along here could be regulated, and basically the gene is still effectively off. So, for example, when FOXO is sequestered in the cytoplasm, it's effectively off. Yep, it's got transcribed, translated, and everything happened, but it still can't do its job. And so we want to be mindful of all the steps in gene expression and how they can be regulated at different points along the way. So that's what this assignment is about, especially focused on inside the cytoplasm. So big distinction, inside the nucleus, big distinction between that and the cytoplasm activity, okay? So RNA transcribed from the transcription unit is called the primary transcript, okay? And how would we say that? Well, here's a gene, here's the transcription unit. The transcription unit is going to be the part of the DNA that gets transcribed by RNA polymerase, and we're going to make an RNA that's the same length of that. So that's going to be the primary transcript. So the transcription unit and the primary transcript are the same length, exactly the same length. The difference is a transcription unit is made of DNA, a primary transcript is made of RNA. And then that primary transcript is going to get modified and processed and leave the nucleus as a mature transcript. Okay, so the RNA transcript to the primary transcript, we have that mature transcript. And so for coding genes, the main types of RNA processing in the nucleus are these. We're going to have a 5' cap added, a poly A tail added, and RNA splicing can occur in some cases. And we're going to be especially focused on mRNA, messenger RNA, coding gene RNA, but non-coding genes also have RNA processing. And so these steps here are we're especially focusing on the mRNA. And non-encoding genes can also have this type of RNA processing, but they also have especially some different types of RNA processing. So here's what this is, and we're going to somewhere it should say that we're going to draw this. So, I have a better drawing of it. But this is just the first one, the cap. So every RNA starts at the 5' end, it goes to the 3' end, and so this is called a 5' cap, and you of course don't need to know the structure or the detailed name of it. It has a G nucleotide bound, covalently bound as the 5' cap, and so some people call it a 5' G. It is just there chemically. So we're going to have a 5' cap. That happens as the primary transcript is getting transcribed. It's going to happen immediately, right? Like that 5' cap on mRNA is a modification, and it especially is important in helping stabilize the mRNA. So it's got to happen to help the RNA from getting degraded. And if RNA doesn't get capped, it's just going to get degraded. So it's just a simple kind of check. It also helps it say, yep, it needs to get that to get out of the nucleus, and it needs that also to bind to ribosome and help with translation. So it's a pretty simple story for the 5' cap. Then the poly A tail is a series of A's, a long series of A's. They can be 100, 200 A's long, and they're added at the end of most mRNAs, and that's called polyadenylation, and it also helps stabilize the mRNA, facilitates its export, and it's really important for translation. So these are the first two steps that happen, and they happen in the nucleus. The 5' cap is added almost immediately, and the poly A tail has happened as soon as the primary transcript is formed. And here's examples of in the 3' end of the primary transcript, there's a signal, a sequence that will guide the polyadenylase, an enzyme that's going to add this poly A tail. And This is just saying that the enzyme is going to find this signal and then find the poly A site, and then it's going to cut the primary transcript and add this long poly A tail. So for us, all we have to do is we have to say that the mRNA gets polyadenylated, and that means it gets poly A tail added. Okay, and that's it. So this is what we should draw. Here is a RNA in the nucleus that's already had its 5' cap added and the poly A tail added. It's a coding gene. So this is the coding region. They're just using a different abbreviation here. Open reading frame, synonymous with coding region. And something we haven't talked about, but just we should be aware of, is that the transcript, even the mature transcript, is longer than the coding region. And this is RNA that's part of the transcript that's called the 5' untranslated region and the 3' untranslated region. And so these are parts of the RNA and parts of the transcription unit, and they just don't get, they're not part of the coding region. So this is the region down here where it has that signal sequence for adding the poly A tail. And this sequence up here is important in helping identify where the ribosome should start. So that's why there's a little shape to this, is because there's some structure to this that's going to help the ribosome. And so ribosomes on the cytoplasm, we're going to see this as an mRNA because it's got a 5' cap, it's got a poly A tail, and then it's also got some 5' untranslated region structure to help guide the ribosome to where to start to add amino acids to a growing protein chain based on this coding region, like that. Okay, so draw that, very simple, and label that. Should be able to do that on an exam as well. And that's part of RNA processing. Okay, and so this just shows out in the cytoplasm that when the mature mRNA in the cytoplasm binds to the ribosome, it actually binds to the ribosome, and that includes proteins that will bind and recognize the 5' cap, and also proteins that will bind the poly A tail. And those are really essential for translation. So this is going to be the ribosome. This is going to be translation out in the cytoplasm. And again, just underscoring that that to properly use an mRNA to guide the synthesis of any protein, the RNA has to have a 5' cap, has to have a poly tail almost always. And then the ribosome is also recognizing this 5' untranslated region to guide it to the coding region, like that. Okay, great. So that's that. And here's just a little information about c-Myc, and it has a poly tail that's really important. And its tail is a long sequence of poly A tails. And then Myc, the poly A tail's length and dynamics are linked both to translation. And so it happens to have a short poly A tail and is unstable. And what happens is with Myc and other polyadenylated proteins is that this tail tends to get chewed up as the RNA is bound to the ribosome. And so this long tail does provide a kind of timer mechanism for how long this RNA is going to be around. And so when we say, oh, it's got a short poly A tail, c-Myc is not going to be around very long. If the tail is real short, the ribosome doesn't even bind to the RNA, and the RNA is pretty stable because it's really only during the translation where the poly A tail gets shortened. Great. All right. And just the paper from where that previous statement about Myc came about. Okay, and so the next thing we want to talk about is the other main type of RNA processing in the nucleus, and that's RNA splicing. And so that's going to look like this, where we have a 5' cap, we have a 3' poly A tail, we have a coding region like this. And what it shows is that in the RNA, and it's not limited to the coding region, it could be outside of the coding region as well, but in the RNA, there are parts of the RNA that get removed. and that's happening as part of RNA processing inside the nucleus. And what gets removed are called introns, and what gets maintained are called exons. And so you can see that this is an essential part of RNA processing. Not all genes have introns, but some do, and the introns get removed. And so this would be an example of a transcript that's already had some processing done. It's still in the nucleus, and it needs to have the RNA introns removed. In general, I mentioned that even before the primary transcript gets finished being made, the 5' cap is added, and as soon as the 3' end is reached, the poly A tail gets added. And so these introns get removed next. It all happens pretty fast inside the nucleus. And so one thing that we could say is this, and let's say that one of the assignments is going to show draw and label an example of alternative splicing. So we'll just skip it a little bit ahead here to put into this drawing. You could do this for your drawing. You start out and say this is DNA, and this is the transcription unit. So we're solid on that. We can draw that all day long. And then here is the primary transcript. Okay, so that's RNA, and that's the primary transcript. And if this gene has introns, there are introns here. I'll draw them like this. And the introns can be found in the DNA and in the primary transcript. And then in the mature transcript, there are no introns. So that's a big distinction between the mature transcript and the primary transcript is the mature transcript has had the introns removed. What remains are these portions, and these portions are called exons. And the exons are simply numbered. So here's exon 1, 2, 3. And if we draw the DNA or the primary transcript, the exons can be interrupted by these introns. And then when we draw the mature RNA, there are no introns. They're just the exons. And so that's really important. The introns cannot code for a protein. The exons code for the protein. And so the introns need to get removed like that. And here's a draw and label, an example of alternative splicing. And I'll loop back and talk about those slides. But here's an example of alternative splicing. Is some genes have regulation so that they make more than one mature transcript from one primary transcript. They can make this mature transcript, or they could make that transcript, where they actually treat exon 2 as an intron and remove all of it. And that splicing can be done alternatively in a variety of different ways. For example, it could be that it saves exons 1 and 2 and throws away 3, like that. Or we'll see alternative splicing can be that there are alternative introns and exons, so that we can include, like it could be a 2A and a 2B, and between them, those exons are introns, and we could do alternative splicing of any combination. The alternative splicing is not random, it's highly regulated. So let's back up and take a look at those. But that would be a good way to draw and label this as part of your assignment, is to draw the DNA, show that it has introns and exons, draw the primary transcripts, introns and exons, draw the mature transcript, no introns. Okay, that's the big distinction there. So let's just look back at this. Introns get removed. That's called RNA splicing. We splice out the introns, and here's a figure of what I just drew. So transcription unit is made of DNA. The primary transcript is sometimes called pre-mRNA. it's exactly the same length as the transcription unit, then splicing occurs, and a mature RNA has no introns, and it has these exons, like that. And when they get translated, those can be included as part of the coding regions, and so you would have a protein that essentially looks like this. And I see if I have it on this slide. Yep. So these are some examples of alternative splicing. We could have this, that's the most obvious one, include all the exons. But we could exclude exon 2 or 4 or 3, like that. And in doing so, it's just showing one protein. But you can imagine over here, the protein could have the same part of the green and the same blue and the same red, and this protein would just be missing part of it. It'd be missing 3. So when we look at alternative splicing, these different forms of the protein have some sequences that are identical and some that are totally different, like that, okay? And these different forms of the protein are called isoforms. So isoforms are different proteins encoded by the same gene. And this is common. In humans, there are many, many genes that are alternatively spliced. And they make multiple protein isoforms. Like, for example, those nuclear receptors, they have multiple isoform proteins. So the estrogen receptor has different forms that are like this, where portions of it are exactly the same. So now we have to make sure we can distinguish this, and that is that alternative splicing leads to very similar proteins that are different in specific ways, and those proteins are called isoforms. And let's just make sure we can distinguish that from paralogs. Remember, paralogs are different genes. Different genes in the same species that are understood to have risen by accidental gene duplication over evolutionary time. So paralogs are different. And it's confusing, but just learn that paralogs are different genes, and protein isoforms are from the same gene through alternative splicing. So we see both of those in humans and other fancy organisms. I mean fancy, more fancy than a little simple worm or a little simple fruit fly. We don't have a lot more genes than those do, and the genes we have are often an example of, oh, there's one of those in a simple worm, and then it gets duplicated. It's a gene family, and then another way of providing variability of similar functions would be alternative splicing, like that. And so that happens a lot, and we just need to know that it's part of RNA processing, that it's regulated. It happens inside the nucleus. What comes out of the nucleus is alternatively spliced products that are regulated and specific for certain proteins. So draw and label an example of that, so we're familiar with that, okay? This just shows that splicing process, and so there's the intron. This is happening inside the nucleus. Uh-oh, I lost my pin function. No, okay, this is the nucleus. There's an exon, there's an intron, there's an exon. And so it's a regulated process of splicing, removing the intron, splicing it out, and there is a complex protein and RNA that binds to these areas and splices out the intron. And that's shown there as a SNRP, small nuclear ribonucleoprotein. So SNRPs are involved in splicing, and that's all we need to know is that they're involved in splicing, and they're made up of proteins plus a non-coding RNA, that's called snRNAs, small nuclear RNAs. There are the names that those are given, and they make up these SNRPs. And so SNRPs are involved in splicing, and they would be somewhat different forms of the SNRPs to do alternative splicing. So you can see it's a pretty complicated structure, and that allows for us to make a variety of SNRPs to do a variety of alternative splicing. Where is it happening? It's all happening inside the nucleus. Like that? Yep. And we'll want to use this example not in greater detail, but just an awareness that splicing occurs in the nucleus, and these proteins of the SNRPs and the non-coding RNA, the snRNA of the SNRPs, need to be in the nucleus to do their work. Great. Okay, and so just showing a little more detail of that, leading to the mature RNA. Great. So RNA process splicing, a key process, gene expression, removes non-coding regions from introns, pre-mRNA, and joins coding region exon to produce mature RNA. And it's mediated by these SNRPs that have small nuclear RNAs and proteins. Okay, they're part of this spliceosome. That's what is regulating splicing. So it's not a random process, it's a regulated process. Okay, and so that's great. That's the center now of our RNA processing assignment, is that these are occurring in the nucleus. And I mentioned that as part of this assignment, I want to just add on this RNA interference activity that occurs primarily outside of the nucleus. So this is outside of the nucleus. It's not RNA processing, but it's the next step where RNAs can be regulated. And so after mature RNA leaves the nucleus, RNA interference can occur. And the RNA that's coming out, we'll stick with it, that it's an mRNA. It's heading for the ribosome to get translated. And there are some specific siRNAs, so we should learn that, siRNA, small interfering RNAs, that have exact matches with part of the target mRNA. So that means that there's an A here, and a T here, and a GC, a G here, a C here, and say another C, and another C. And this matching is exact, and it's about 20 nucleotides long. And so the siRNAs are short RNAs that have an exact match with the target, and they bind, and they have this double-stranded RNA, and it causes this target mRNA to get degraded. So it just gets destroyed. Okay? And that happens in nature, and it happens in plants and animals. And these small interfering RNAs, well, they must have their own gene in the nucleus, and they must get transcribed as a non-coding gene, and they must get prepared to do their job. And they do. And so part of their processing is different that we've learned about, because part of their processing involves cutting this longer RNA to make this smaller, shorter, small interfering RNA. And so we're not emphasizing that processing. I'm just using that example to say that for non-coding genes, the processing can be different than for coding genes. Coding genes really emphasize the 5' cap, the poly A tail, and the splicing. Okay, so this is out in the nucleus after RNA processing is done, and one thing that happen is a small interfering RNA can bind to the target mRNA and cause it to be degraded, and that turns the gene off. And in doing so, we say we knock down gene expression. And what knockdown means is we're going to decrease gene expression. So these siRNAs are always involved in decreasing the gene expression of target RNAs. When people talk about knockdowns, they like to compare a knockdown with, in the nucleus, we can have a null allele. And a null allele has 0% activity. And we could use the term, because it's another official term, knockout. And that's from boxing, fighting. And a knockout is when the other your opponent is knocked out and it's done. There's nothing you can do. It can't get back up and fight. It's done. It's over. And that's what happens with an allele. It can never be fixed. It's gone for good, right? The DNA has been changed permanently. For a knockdown, if we remove the small interfering RNA, so if we just decrease the expression of the small interfering RNA, the mRNA will no longer get degraded. It will not be knocked down. It'll be like a fighter that got knocked down and can get back up. So just a couple of terms, and they are official terms. A knockout happens in the DNA, and these small interfering RNAs cause RNA interference, RNAi, by knocking down gene expression. Temporarily, as long as the siRNA is there, the RNA will get degraded and the gene will be decreased. So it's always a decrease, like that the RNA interference decrease. Okay? All right. And so here's just a little look of that. We start out with a longer RNA. It gets processed in the nucleus to a shorter RNA, and then it binds to a target, and it cuts up the target gene. So siRNA are involved in RNA interference, decreasing gene expression of target RNAs by having the RNA get degraded in the cytoplasm. Okay, so just a little way of helping remember this. Scientists have known about this for a long time, 30 years like that. These two scientists won the Nobel Prize for this. This kind of RNA involves small interfering RNA, and it causes the RNA to be knocked down. This process is natural in all plants and animals. It occurs. Some plants and animals have more of it than others, like that, but definitely, present in all animals, present in humans. The real reason that these scientists won the Nobel Prize is that scientists realized that they could actually, in a test tube, a custom siRNA gene could be designed and used in experiments. So, for example, you could, say, be studying insulin and in a mouse, and you could say, geez, we want to look at mutations in the gene, but that takes a lot of work. Well, now we could just find the insulin gene sequence and find where the RNA transcript is, the transcript that's going to be, part of the RNA, and then just design a short interfering RNA gene that has the sequence that would target insulin. And in an experiment, we could add our new gene to the cells, and they would knock down insulin gene expression. They wouldn't destroy the gene. They wouldn't have to go through all the work of, finding mutations, destroying the gene. We could just target its destruction. So the scientists realized that even though this is a natural process, we could take advantage of it in experiments. And so that's a big reason why these scientists won the Nobel Prize. And they won it when they were young. They'd done the work recently because it was exciting. And it's natural, but it also provides opportunity to do experiments on any gene we want, really in any species. So a really powerful opportunity. We don't need to learn the details of these steps, but just the idea that that siRNAs come from a non-coding gene, and they get transcribed, and they go through their own preparation, RNA processing. You can see there's no 5' cap, there's no poly A tail, there's no intron splicing here. That's mostly focused on the mRNA, the coding gene RNAs. Okay, so there's another kind of RNA called microRNA. And microRNAs were discovered at the same time as siRNAs, but they work differently. So microRNAs or miRNAs are also look really similar. They're short RNAs that can base pair with a target gene, a target mRNA. And there's some really subtle differences between microRNAs and siRNAs, and they're both involved in RNAi, RNA interference, knocking down gene expression by affecting the RNA in the cytoplasm. And in general, siRNAs work by degrading the target, and mRNAs work generally by blocking translation. So the RNA is still there, but the ribosome just can't use it. Same effective result. We could degrade the RNA, we could block it from translation. Same effective result. We are going to knock down gene expression. Okay? And so I want to add that here too, to kind of compare and contrast this. This is happening inside the nucleus. This is primarily for mRNAs. Processing of non-coding RNAs also occurs in the nucleus. That RNA processing is not the same. There's some similarities, some big differences like that. And examples of non-coding RNAs I mentioned before, well, transfer RNA, tRNA, ribosomal RNA, snRNAs, small nuclear RNAs. We just learned about those. They're in SNRPs. They're involved in splicing, so this may be something new to you, right? And these are non-coding. And then we just learned about siRNAs. And then microRNAs, miRNAs. And they are also non-coding RNAs from non-coding genes, and these are involved in RNA interference, knocking down gene expression by dealing with the RNAs. Okay, great. And so this just shows the bottom of a bigger figure I want to look at to just show you that this is what siRNAs do. They destroy the RNA, and this is what miRNAs do, and they block translation. Okay, and I'll just show you this, that there's some pretty subtle differences between microRNAs and interfering RNAs, and it took scientists several years to figure that out. And so here's just an example of this. They're really similar in their RNA processing, in preparing the RNAs to be ready to do their job. But ultimately, the siRNAs are really more involved in degrading the target, and the mRNAs are involved in blocking the translation of the target. One way to help remember that is this, and this is just very surprising, and it is that back, 20 years ago, these two scientists won the Nobel Prize, and they were studying a worm, a nematode, that's a model organism like a fruit fly. They were studying the nematode, and they were studying how the nematode grows up from a baby to an adult nematode, and they found genes that were regulated by RNA interference using siRNAs. And scientists realized, we could use these in an experiment. And so it was very exciting because we could use these to do a lot more science beyond just what is found in nature. It's a good example of molecular biologists using a discovery and turning it into a tool, like how we saw early in our course. See this glowing protein from jellyfish? Well, we could put the gene in any organism and make specific cells glow. So another good example of that. Then these scientists were studying that same process at the same time, also, weirdly, surprisingly, in nematodes. And it was quite startling when these scientists won the Nobel Prize, because these scientists had actually done their work a little bit earlier. And even at the level of, Nobel Prizes over the last 20 years, there was confusion, even among the scientists, about why did these scientists win and not these. And it wasn't even clear at the time, but there was sort of hints that this was a different story. These scientists were studying the microRNAs, miRNAs, and they don't work by destroying the RNA, they block translation, like that. And so these scientists won the Nobel Prize first. It turns out that both processes are found in plants and animals. This is rare in nature. This is common in nature. And so it's just really kind of an interesting and memorable thing that both sets of scientists won the Nobel Prize for RNA interference. It's a big deal in nature. And the first scientists that won, though, were the ones who were like, whoa, we can use this in an experiment. And it's good for an experiment because it's easy to test if the RNA can be detected. There are lots of methods that can detect RNA. And when you knock down gene expression using siRNAs, you can test and see, do we still see the RNA? Yes or no? If no, knock down. It worked. Our experiment worked. On the other hand, these microRNAs don't cause the destruction of the target RNA, so they're not as easy to use in experiments, even though they are more common in nature. Great. Okay? And so a little bit about microRNA. They silence mRNAs by one or more of the following mechanisms. They can cut it, but people more often want to emphasize they reduce translation like that. That's the dominant thing. So it's not a perfect distinction, and that's why, scientists were a little bit confused over the years by it. But now people are seeing more and more that we want to distinguish between the miRNA, microRNAs, and RNA interference by blocking primarily translation versus the siRNAs, small interfering RNAs, that cause RNA interference by causing degradation of target mRNAs. Great. Okay. So just a little bit about that you can read through just to see that. And just a little bit more leading to that distinction like that. Okay. And so I think that we want to then draw a picture of this. Okay. And I won't draw this out because it's getting to be a little bit of a long video, but you can do that based on what you learned, right? Of whether they get knocked down by destruction, degradation of the target mRNAs, or by blocking translation, by blocking the RNAs used by the ribosome. Both of those occur. The drawing would be to be in the cytoplasm. And this is not RNA processing. RNA processing takes place in the nucleus. So after the mature RNA comes out, pretty quickly has the situation where it could be affected by RNAi. So these are all good labels to add to this drawing. And I think, just a little bit more here and a little bit more information about non-coding genes, and they have RNA processing. And they occurs in these non-coding RNAs, and the non-coding RNAs are, again, small interfering RNA and microRNA, and so some of this RNA processing actually is really significant because the non-coding RNAs, that will be the functional product of the gene. So we're going to need to take the non-coding RNAs, and they're going to make a primary transcript, but it's not ready to do its final job. So with the small interfering RNAs, microRNAs, they're going to get cut up and prepared to do their job. And here's just a couple of examples of that. So tRNAs are non-coding RNAs, and they're transfer RNAs. They're bringing the amino acid to the ribosome for the next amino acid in the protein growing chain. And this is just an indication to see that here's the RNA, and this is showing all of the covalent modifications that occur. So to get a mature tRNA that leaves the nucleus, we're not going to do splicing, we're not going to do 5' cap, poly A tail, that's almost entirely mRNAs. For these non-coding RNAs, we have a variety of other kinds of modifications, and there can be a lot because we have to make the final functional protein. So we don't need to learn these names in detail, but just the idea that tRNA goes through a maturation process in the nucleus and RNA processing in the nucleus. That's different. Now, It turns out that when you look at RNAs, these aren't perfect distinctions. For example, some mRNAs also can have some modifications like this. They're specific, and we're not going to worry about any specific examples. Okay? And so just this idea of RNA processing happening in the nucleus, and there's lots of stuff. And we want to learn about the nucleus and what happens in there. And before these can leave through the nuclear pore, so before they can have exportin protein help them get out of the nucleus, they need to be mature RNAs. And proteins bound to those RNAs will signal whether the RNA is ready to leave the nucleus yet or not. And so just to say again, that there's a variety of modifications. Some messenger RNAs also have those found in transfer RNA and other RNAs, but it's rare, and we're not going to learn any specific examples. Just to know there's lots of processing, and it's a step in the pathway of gene expression. Okay, great. So I put this here at the end as well, but it's a drawing we already did. So if you didn't do that drawing at the start, do it again, or look back at it, see if there's anything you want to add to that original drawing that we did. We did talk initially about RNA interference. We want to make sure that's included in that drawing. Okay, great. And that's where we're at for this assignment. Awesome. Thanks.